Hello and welcome to another fresh episode of Bioenigma Uncovered. In our ongoing QIIME2 series, we have so far learned how to download and install QIIME2 environment into your system and how to import FASTQ data into your QIIME2 environment, followed by how to perform demultiplexing and denoising using DADA tool into QIIME2. And in addition, we have also learned how to download sample metadata files and validate them using the QIIME2's QIIME tool. In this video, we are going to learn about how to perform taxonomic classification using QIIME2. So without any further delay, let's start today's video. For taxonomic classification in QIIME2, we require two important things. First is a set of representative sequences which are in the form of rep-seqs.qza file. This file contains numerous sequences which contain a unique set of ASVs or amplicon sequence variants. In addition to this file, we also require a reference database classifier. For the help of students and researchers, QIIME2 provides a pre-trained classifier which have been already matched against various hypervariable regions of 16S ribosomal rRNA. These classifiers can be provided by various tools such as Silva, GreenGenes2 or GTDB. Specifically in case of QIIME2, the reference database classifiers are provided by Silva. So these Silva based classifiers, they can match the hypervariable regions such as V3, V4 or a mixture of V3, V4 regions. In particular, this tool, we are going to use the reference database classifier, that is Silva-138-99-V3 v4-classifier.qza file, as you can see on your screen. So using a region matched classifier improves first the taxonomic accuracy while also avoiding any misclassification which can reduce the accuracy of our data analysis in the subsequent steps. And now, as you can see on your screen, we are going to run the step of taxonomic classification. An important point that you must know before running this step is that this particular exercise is memory intensive, and therefore you require a very powerful system for running this program or running this particular part. Otherwise, you will see that your system is going to repeatedly crash and you are going to get no result, which can be very frustrating for the students. So personally, we have faced a lot of issues. So what I suggest is you need to use a powerful workstation which is having at least 64 GB of RAM. Besides, if you are using a WSL or Windows subsystem for Linux, or even you are working in a Linux environment, then your system must have an empty RAM, empty space of like 100 or more than 100 GB. As you can see on your screen, we are running this command QIIME feature dash classifier space classify dash sklearn, followed by two input commands. The first input command, as you can see on your screen, requires the execution or requires the input of your database classifier, reference database classifier in the form of Silva. Or in the next step, you require is the input of your input files or input sequences, which is in the form of rep-seqs.qza. This will give you, executing this command will give you an output in the form of taxonomy.qza file. As you can see on your screen, the command is being executed. One important thing that you must know is that the QIIME2 is going to analyze each of your ASV and compare it against the reference database, and each ASV will be provided a taxonomic hierarchy right from kingdom to the species level, provided that the correct information or the relevant information is already present for that particular sequence in the reference database. In the next step, we are going to convert this taxonomy.qza file into a visualization file or that is .qzv file, which can be viewed or analyzed using the QIIME2 .view.org program or the website. So this .qzv file can now be opened into view.QIIME2.org website for interpretation. Into your browser web page, type view.QIIME2.org as you can see on your screen. So the layout of this QIIME2.org is like this. Click on this drag and drop or click here option. Go to your desired folder where your file has been saved. So as you can see on your screen, I just click on the taxonomy.qzv file, click on open, and you will see a layout like this. So this taxonomy.qzv file has three columns. The first column is feature ID, the second column is taxon, and the third is the confidence level. Feature ID is basically the serial number or the specific identity number provided to a particular sequence or to the particular ASV. Taxon consists various taxonomic hierarchies such as division, phylum, class, order, family, genus, species, and so on. So for various sequence IDs, you are going to see various types of taxons. In the third column, the confidence number or the confidence score is provided. A higher confidence score indicates that the particular ID with the specific taxon or the taxonomic hierarchy is present at a desirable certainty. As the confidence score goes on, the uncertainty in terms of these mentioned taxon, it goes on increasing, or the certainty of these taxon goes on decreasing. So you can see that majority of these IDs contains the taxons at a higher confidence levels. Means these taxons are certainly going to be present in this particular rep-seqs.qzv file. So that's it for today's video. In the next video, we are going to learn about how to perform alpha and beta diversity analysis using the QIIME2 tool. If you do end up like our content, then please don't forget to hit the bell icon, and also please do subscribe our channel. I'll meet you soon in another video. Till then, take care and goodbye. Thank you so much for watching this video.